Bismillahirrahmanirrahim. Today I will discuss the development of CNS parts of brain, and especially we will discuss today the development of the spinal cord. The today's objectives are the formation of neural tube, brain development in general features, spinal cord development, cervical flexures and the cephalic flexures. In next lecture we will discuss the positional changes of cord, pituitary gland, abnormalities of the CNS. Little bit abnormalities I will discuss today, but in detail I will give you another lecture. This is the diagram. This diagram shows the presomoid embryo at the 18th day. This is 18th day. There is no presence of somites. In next diagram you can see this, there is presence of 1, 2, 3 somites. The somites will appear at the 20th day of development, and in each day 3 to 4 somites are formed. So the neural tube is clearly visible with this blue in color. In this section, this is the neural tube and these are the somites. Now in next slide, you can see this blue color is the surface ectoderm and this is the mesoderm, intraembryonic mesoderm, and this yellow color line indicates the endoderm. In the surface ectoderm, in front of this notochord, the surface ectoderm becomes thickened and it will give rise the neural plate. Then this neural plate forms a groove which is known as the neural groove. And after that, when this neural groove becomes neural tube, these neural crest cells shift. In this diagram you can see that these neural crest cells are going to shift different parts of the body for the development of the different structures. In this diagram you can see these neural crest cells are going to shift in the developing suprarenal gland, and these neural crest cells give the medulla of this suprarenal gland. And this is the GIT, because the yolk sac give rise the GIT, and these neural crest cells give rise the GIT ganglia. Now in this diagram the neural tube has closed. So in next slide you can see these are the somites, and these are now the neural tube starts to close, and this is the day of the 22nd day. And I have told you that the somites appear on the first pair of somites appear to develop at the 20th day of the development, and this is the 22nd day. So there should be the 6 to 12 somites should be present. 7 distinct somites are visible on each side. These are 7 and these are the 7 on each side of this is blue color is the neural tube. In diagram B, dorsal view of human embryo at approximately 23rd day. The nervous system is in connection. ये जो opening है, these connections, that is the anterior neuropore and this is the posterior neuropores. They are continuous with the amniotic cavity through these canals, cranial and the caudal neuropores. This cranial neuropore is also known as the anterior neuropore, and while this caudal neuropore is also known as the posterior neuropore. So the central nervous system appear at the beginning of the 3rd week. This is the beginning of the 3rd week. So the neurulation starts to appear at the 3rd week. This blue color is the ectoderm. In the ectoderm there is the thickening of the ectoderm which gives rise the neural plate. In next diagram, the neural plate going to form the folds. These folds this fold, the formation of these fold is the second step for the in the stages of development of new relation. And in the third diagram, these neural folds are now going to fuse and forms the neural tube. With further development, the neural fold continues to, this is the, these are the neural groove and neural fold, these are the neural crests. Neural crests, then they come together in the midline and fuse to form the neural tube. Now in the central nervous system, the fusion begins in the cervical region. It means the cranial neuropore, which is also known as the anterior neuropore, grows first. That is the 25th day of the development. And the posterior neuropore closes 2 days later, 2 or 3 days later. It means 27th or 28th days of the development. Once the fusion is initiated, the open end of the neural tube, these tubes, form the cranial and the caudal neuropores that communicate with the overlying amniotic cavity. So the anterior neuropore will close at the 25th day of the development, while the posterior neuropore closes at the 27th or 28th day of the development. In this section, when the embryo goes to folding, that is known as the head and tail folding or the cephalocaudal folding, these neural tube give rise three primary brain vesicles: forebrain, midbrain, and hindbrain. The forebrain is also known as the prosencephalon, midbrain is known as the mesencephalon, while the hindbrain is known as the rhombencephalon. So the cephalic end of the neural tube shows three dilatations. These dilatations appear in the cephalic region. The primary brain, these are known as the primary brain vesicles. It is very important question for the exam point of view. They will ask mostly write down the name of the primary brain vesicles. The answer is the prosencephalon, mesencephalon, and the rhombencephalon. Simultaneously, it forms the two flexures. You can see this is one flexure and this is the another flexure. This is the cephalic flexure and this is the cervical flexure. Mostly they give a basic cue that cervical flexure appear between, this is the spinal cord, and above the spinal cord should be the hindbrain. So the cervical flexure is present at the junction of the hindbrain and the spinal cord. It is very important basic cue. And in the cephalic flexure, this is the cephalic flexure. It is present in the midbrain region. Present in the midbrain region. Now these are the parts of the brain which are showing the primary brain vesicles, which give rise to different parts of the brain. This is this telencephalon and diencephalon are the parts of the prosencephalon. The prosencephalon divided into telencephalon and diencephalon. Telencephalon shows the two outpocketing, and these will give rise to cerebral hemisphere, and the cavity of the cerebral hemisphere are the lateral ventricles, two lateral ventricles. So the telencephalon give rise cerebral hemisphere, while the diencephalon characterized by the outgrowth of the optic vesicle. Optic vesicle will give rise initiate the formation of the eye. In this diagram, this is the neural tube. These are the primary brain vesicles: prosencephalon, mesencephalon, and rhombencephalon. What are the derivatives of the prosencephalon? Telen and diencephalon. Mesencephalon, throughout it remains mesencephalon, which gives rise to midbrain, and the rhombencephalon is divided into metin and myelencephalon. There is an isthmus between these metin and myelencephalon. So the telencephalon gives rise cerebral hemisphere, while the diencephalon gives rise thalamus and hypothalamus. Mesencephalon cephalon give rise midbrain. Metencephalon give rise pons and cerebellum. It is very, very, very important BCQ. While the myelencephalon give rise the medulla oblongata. So the brain stem consists of midbrain, pons, and medulla, while the hindbrain consists of pons, cerebellum, and medulla oblongata. The midbrain is not included in hindbrain. It is very, very, very important. What is the cavity of the cerebellum hemisphere? Lateral ventricle. Cavity of the dancephalon, mean thalamus and hypothalamus, is the third ventricle. Cavity of midbrain is the cerebral aqueduct, which is also known as the ductus salvius. And the cavity of the pons and medulla is the fourth ventricle. Cavity of the spinal cord is the central canal. It is very important tabulated form. These are the vesicles which are showing vesicles and the flexure between these vesicles. Metin and myelin, one flexure is present over here. Now the development of the brain, this is the rhombencephalon, mean the hind brain consists of two parts, metin and myelin. Metin and myelin. This is metin and this is the myelin. The metencephalon, this metencephalon give rise pons and cerebellum, while the myelencephalon give rise the medulla, and the flexure between these two part is known as the pontine flexure. Is known as pontine flexure. This is the epithelium. This epithelium is initially, you can see this is the epithelium. Each cavity, I have told you already in my previous lectures, the cavity of the brain, mean ventricles of the brain, they are lined by the ependymal cells which are columnar in shape. These ependymal cells will proliferate and give rise is pseudostratified type epithelium. These cells are now known as the neuroepithelial cells. The next question asked in the examination is name the neuroepithelial cells. These are the neuroepithelial cells. First of all, the neuroepithelial cells give rise the glioblast. These are, and there is the ependymal cells. Ependymal cells proliferate and give rise the pseudostratified type of cells, and these cells, neuroepithelial cells, which are known as the neuroepithelial cells, give rise glioblast give rise the astrocytes, which are of two types, the protoplasmic and the fibrillar. They also give rise bipolar neuron, multipolar neuron, and the lastly mesenchymal cells give rise the oligodendrocytes and macroglial cells. It is very important cell. These cells are derived from the ectodermal in origin, while this mesenchymal cells which give rise the microglial cells, they act as the macrophages of the brain, because the macrophages of the brain are known as the macroglia and they are mesodermal in origin, while all other cells are ectodermal in origin. It is very important question as exam point of view. Now in this diagram you can see the stages of development of the spinal cord. Now we are going to discuss the development of the spinal cord. Formation of the motor and dense sensory, or how they form and how this intermediate horn or column is formed. Now first we will discuss this diagram, the neuroepithelial cells. These are the neuroepithelial cells or the neuroblast cells. They forms the mental layer. This is the mental layer which is darker. A zone around this neuroepithelial layer, and it is known as the mental layer. Known as mental layer. And the layer present outside the mental layer, which is lighter in color, is known as the marginal layer. This mental layer give rise gray matter, while this marginal layer give rise the white matter. It is also an important OSPI question or an VCQ. Now, as a result of continuous addition of the neuroepithelial cells in the mental layer, this is the mental layer.